Hey biology students, I'm Miss Garibaldi and we have
Hi, Miss Patterson.
and we're going to go over how to create a wet mount of an onion slide for your plant cell. And so we're going to go over what materials you're going to need for your lab first, and then we will demonstrate how to do a proper wet mount.
All right. We have our slide, just our microscope slide. And then we have a cover slip that goes over your specimen. We have a toothpick, which we'll explain in just a second. And we have methylene blue stain for you to actually be able to see what you're looking at. So when we do the onion, you'll stain it so that you can see the cells. We have a peach here. That's an example.
Yeah, for this purpose of this video, we're going to be using a peach. But you will have onion, we'll cut it up for you. We'll just make sure you keep the lid on because it can get pretty stinky when you're doing your plant cell slide. So the first thing to remember when you're using the microscope, and this may seem like an obvious thing, but you need to make sure it's plugged in and that it's turned on. The on switch is towards the right. Some of the microscopes may take a little while before they turn on.
Yeah, please came on.
Okay.
Yeah, wrong.
All right, so the next step with the microscope, when you're using the microscope, there's a few knobs and structures that you will need to be familiar with. The first one is this large knob here. That's the coarse adjustment knob. And then we have the smaller knob, which is a fine adjustment knob. So the coarse adjustment knob moves the stage up and down, where the fine adjustment knob will focus in on the specimen. So we always want to remember to use the coarse adjustment first, and then once you actually can see something in the eyepiece of your microscope, you then will use your fine adjustment knob to focus in.
And then these. You also want to make sure that you're on the lowest objective, which is the 4x magnification, so it's going to be the shortest. And then once you get it in focus on that, you can start to turn to your 10, and then maybe make some adjustments, and then your 40. So your 40 is going to be only used at the very end once you have it in focus under 10 and 4, or else it's going to be really hard to find the cells in the field of view. So the first one, we have a bacterial cell that's prepared slide. So we will put that on our stage. And you can see that there's a little round circle where the specimen is, so you're going to want to make sure that's over the light. So you may have to do some adjustments to the stage. So you can pull it back and forth. And then there's a knob on the side to move the stage so that the slide comes in view. So now that I have it over the light, I'm going to start moving the coarse adjustment knob. And this one you want to move pretty slow, but it's going to take a while for it to come into focus. So there, that took us about the same amount of time, right? And then you can move it, the fine adjustment knob, to clear up the picture a little bit. But again, these are very, very sensitive, so don't move them fast at all. And then now that we found it under 4, we can move to 10. And then you may have to make some adjustments with your course, but again, it needs to be really slow. Make sure everyone in your group sees it at different